Thank you for your kind introduction. My name is Jonghyun from South Korea, and thank you for giving me this opportunity to talk about our work titled Tracking Tumorigenesis and the Transition State through Copy Number Variation-Based Pseudotime. As most of you are already aware, one of the ways to tell tumor cells from the normal cells is the copy number variation or the aneuploidy. So if you can see the figure, if you do the single cell analysis, you do the clustering, you get the marker genes. Conventionally, what you get, especially from the breast cells, is that all cells have some characteristics of the normal cell characteristics here. For example, progenitor cells, mature luminal cells, or myoepithelial cells. However, if you run the copy number inference and lay on top of it, you can see one of the cluster is clearly aneuploid over the diploid state. Therefore, one of them is tumor and the other one is not. There are many copy number inference algorithms, most famous being InferCMB, and there are other generations such as copycat and Scaban. When we do these analysis, we see that tumor cells do have these copy number accumulations. And we were wondering if these copy number accumulations could have happened already before the tumorigenesis. And we, based on this hypothesis, on two observations. First one is that the tumor cells, within the tumor cells, there are subclones, and some tumor cells seems to have less CMBs than the others. And from even the normal states, that there are some cells that does appear to have CMB that are carried over to the tumor states. So we wanted to trace this based on the CMB accumulations to see if we can connect the dot from the normal cells to the tumor cells. These are our workflow. We get the single cell data from the cancer cell data. We do the analysis and we isolate the epithelial cells and the tumor cells and annotate it. We get the CMB inference algorithm and CMB metrics, and we apply what's called the non-negative matrix factorization and try to figure out which CMBs are accumulated in the normal and which ones are more important, so to say. Then using this information, we run the neighbor joining methods to try to reconstruct the phylogeny. We ran this on two separate data. One is the mouse data and the other one is the human data. The main difference is that the mouse data has an advantage of tracking from the healthy mouse until the tumor is developed, while the human sample is generally tumor is fully developed and you're trying to sort of retrospectively figure out how this could have happened. So if you see in this data, the main big metrics is the CMV metrics. The gain CMV is red and the loss is blue. And on the top of the column, I have annotated the cell type to the luminal to the tumor. What you see here is that the luminal cells have less CMV. And to the far right, the tumor cells have a lot of CMVs. However, when we do the clustering, then there's this middle cluster of the column where the luminal and the tumor cells are mixed. And if you go all the way down and if you look at the CMVs accumulation, they seem to share the similar CMV patterns of the tumor clusters. Using this information, we ran the phylogenetic tree analysis. And we see, so this is the tree generated with the neighbor joining methods. From the bottom to the top, the cells are aligned, and there is a luminal to the tumor. To the right, the matrix is each cell's corresponding CMV profiles. From the bottom, you see that the luminal cells go from the no CMV accumulation to the top, where tumor, there's a lot of CMV accumulations. However, the transition is not as clear-cut. So there are a lot of mixtures between the normal cells and the tumor cells within these trees. So this gave us enough to try to do this on the human data. And what we see directly compare from the mouse data, the human cells, a lot more of them are tumor cells and a lot more CMVs accumulated. However, we still see the similar pattern of the second middle cluster of the column, that the normal cells and the tumor cells are mixed, and the CMV profile is also similar. We apply the same technique and try to figure out the phylogeny. This time you do see a clear separation, but as I have put in a red box, the separation of the tumor clades begins with the normal cells. And if we zoom in a little bit closer, there is a first CMV gain that is shared with the subset of the normal cells. And maybe these are possibly the precursor event of the tumorigenesis that could have initiated some of that these normal cells are more likely to become tumors than the others. So how those were the inferences based on just a purely CMV. Then how does the expressions differ? Maybe they have specific characteristics that we could define it, use the microgenes to maybe use the preventive measures to stop cancer from occurring altogether. So we define those clades, we term them as a transition state, and I didn't do that. We lay on top of the expressions based on PCA. And what we see here is that the transition states are between the normal cells and the tumor cells. We dug a little bit deeper into this particular CMV, and the CMV gain contained 151 genes, which one of them included the IL-6, which was already known for the breast cancer to cause growth and metastasis. We also ran enrichment analysis, and these genes are involved in functions such as immune modulation, energy generation, and angiogenesis. And possibly these are the factors that initiated the normal cells into becoming a tumor cell. So in summary, we use two techniques, the non-negative matrix factorization and neighbor joining methods, to try to figure out if we could identify possible precursor events from normal cells to become tumor cells. From the one tumor sample data, we have identified the transition states that the normal cells do share some of the CMV events from the tumor cells. And proper identification of these transition states has implications in cancer preventive measures and personalized medicine. So what we really wanted to do is that, as the first presentation mentioned, the proper phylogeny inference, and we would really like to involve the SMVs into this to try to really figure out how this happened. We have defined the transition state in this study for now, and we haven't truly looked at how the expression there are different. So identification of marker genes or arrangement scores, those would be very useful in further investigating what kind of characteristics they would have. And we have chosen breast cancer because breast cancer is known to accumulate a lot of CMVs, and we wanted to see if this could be applied to other cancer types, and those are still ongoing. These are the references we've utilized. These are funding, and I would especially like to thank the EvolCompGen for helping me to get here financially. Thank you very much.
Hello, thank you for the amazing talk. You used the neighbor joining to build a lineage tree using the CMVs. I'm wondering if you can use this tree to analyze the ancestral cells CMV and changes during divisions, and what's your do you have more interpretation about the hidden nodes on the tree?
Thank you for your question. So we have tried a couple of things, but basic assumption, especially with the CMV, is that I assume that normal cells are deployed. So I sort of try to force it so it starts from the clean state. I have tried to figure out if we could start sort of the CMV accumulated sites to try to figure out if there were original cells or stuff, but the trees didn't give a shape that I could interpret. So that was a little bit difficult one. I am trying also with the maximum likelihood and maximum, the other one, parsimony. And those we are also trying to figure out the best method.
I think it would be a very interesting topic to try to come up with a likelihood that explains the CMV changes on in the context of cell lineage, and that would bring us more amazing analysis. And I think there's a huge potential. And
I agree. Thank you very much.